There you go.
Great, thank you. No, as I say, thank you very much for joining the meeting. Thank you all very much for joining up as route rangers as well. I'm really, really excited about this year because we've got a fantastic bunch of farmers who are taking part in the project with us. I'm going to, so we're going to have, as I say, we're going to have a couple of presentations and then there's time for questions at the end, and we're planning to close at about 9:30. We won't do any introductions to start with. It would just take too long to go through everybody, if that's okay. But do use the chat to exchange messages. As long as it's not rude things about me. And then we'll have a question and answer session at the end, and we'll bring everyone in who wants to ask questions. The way to do that, by the way, would be to raise a hand, and we'll come to you in order. I've got just going to share my screen. Okay, how to do it? Excuse me. Hope everyone can see that. So the project, the Truth Project, that's Thriving Roots Underpinning Total Soil Health. It's a £1 million 3-year project, DEFRA funded, and we have 5 industry partners on the project. Just to run through the agenda for the day quickly, I'm going to go spend about 10 minutes talking through the project, give a bit of an overview, and also focus on what we're planning to do in the year 2 trials. We're now currently in year 2. Then I'm going to hand over to Emil Chappelhow at UK Agritech Centre, who's going to do a brief roundup of the year 1 trials. We're then going to hand over to Charlotte White from ADAS, who's going to talk us through Shovelomics. And this is really a key part of one of the key parts of what we're aiming to do with the project. Then, as I say, we'll have a question and answer session at the end, and we're aiming to close at about 9:30. So just briefly about Truth itself, this has come about from a need to understand more about our soils. We know so much about what grows above the ground, but we know precious little about what grows below the ground. And importantly, it's about the data that we want to gather about the soil communities, what's going on below ground. We all know that soil is an incredibly important part of the systems that we manage and that we operate, but we don't actually know exactly how that happens. We don't know how to quantify it yet. But this question of soil health, what is soil health? So that's what we're aiming, and also to understand the relationship between the inputs that we apply and our roots and our soils as well. And not just the inputs, the varieties that we put in the ground and what they're doing, the crop traits involved in that. And we've got some pretty groundbreaking stuff that we're going to be hopefully delivering during this project. So these are the partners involved: University of Nottingham, PES. We've also got John Innes Centre and UK Agritech Centre, as I say. We're also bringing on board ADAS, particularly with their agronomics, and of course Charlotte White, who's going to be guiding us through Shovelomics to help us with that. The 2 main outputs that we're looking to deliver from the project is a robust, practical on-farm platform, we're calling it, which is you, the route rangers. This is about a team of farmers who know more than anyone else in the country, we hope, by the end of this project, about their soils, and can use that knowledge to help others and to bring on knowledge about soils together. And also a knowledge forum to develop the root growth and how best to influence that. And that's the soil circle. And you lie absolutely at the center of the soil circle. You are the ones who will, we hope, gain the most knowledge from this. And we are hoping that you will use the channels that we're providing for you and your own channels to communicate that out to other farmers particularly in the Soil Circle, which is a community of about 350 or so farmers that we have brought together, and then wider out into the innovation community, so that the practices that you see first on farm, that as we gather understandings of the soil and the relationship it has with our crops, that comes out into the innovation community. And as I say, there are various channels that we are providing to do that. Most significantly, the Soil Circle, which is the forum on the Farming Forum, and it is very much, part of the role as a route ranger to get that discussion going. It has to be said, there isn't as much discussion on there as we would like, really, and it really is your job to drive that discussion, bring some information on there, and get the discussion going. And any observations, any results that you have, we'll be posting results on there as well, as we already have. But it's the discussion around that is really the value here, and the knowledge sharing is the important part of this project. We're also attending all sorts of events. I'll give a quick roundup of what events we're going to be attending at the end. But that's a great opportunity, to come together to talk about the projects, to meet people involved, and so on. And there are going to be, as I say, various opportunities to do that over the year. We're also hoping that people will take part in what we're calling Innovation and Action Days. So host their own mini events on their farm, be that a farm walk with just a few local farmers, or you could have a little bit more of a mini event if you like, and we can help and support you in that. And that would be an opportunity to bring together some of your local farmers, talk about what you're doing within the project. It doesn't have to be just within the project, it can be whatever else you want to talk about on the farm, and we can help and support you with that. And a number of you are getting involved in ADAS agronomics and having a little bit more of a deep dive into your trials this year, which is really great to see. So we'll have some results of that later on in the year, I hope. And that'll be a great learning opportunity. Some of you are also involved in the YEN, ADAS YEN as well, and looking again in more detail at how the crop above the ground, how the crop below the ground is affecting growth above the ground. And we'll get some good detailed data on that. So that will be a good aspect. And of course, there's the Shovelomics thing. Now, an important aspect of Shovelomics is so the detailed data that you will get from the analysis that you're going to be providing through the samples, that's exclusive to you. There's no way every farmer in the country can get that information. But at the same time, what we are trying to develop are quick and easy ways where you can accurately assess the health of your crop and more importantly, the roots and your soil. A quick and easy in-field methods to do that. That's what Shovelomics is all about. And with Shovelomics, we're particularly interested in the roots. And so you will get an idea from the soil cores what your roots look like. You'll get an idea from Shovelomics how to measure it as well. And that information is exclusive to you. But other farmers will be able to do the Shovelomics method. So we want to develop that as a farmer-friendly, quick and easy, but more importantly, accurate tool of assessing what your root health looks like. So the Root Ranger role this year, those who were involved last year will remember that there are three main things that you measure in the soil, in your field. So this year, you'll be taking 5 root core samples around the field. There'll be 20 rhizosphere samples, and that will be taken at 2 different stages. So that'll be just before your second fertilizer application and then just after. And we're particularly interested in the rhizosphere. So this is the sheath of soil that clusters around the root. And those are the samples that you'll be taking. And then also 8 dry root samples. This is for our vascular mycorrhizal fungi analysis. And there will be full guidance on how to gather these samples and how to prepare the samples and then send them in. And you'll get a full pack on that. We're also going to be doing 10 soil analysis samples. They're just simple soil analysis samples as well. Some of you will have had your farms mapped with TerraMap, which essentially gives the same information, but we're looking to get the soil samples. And the idea is to do those samples at specific locations in the field that you come back to with your second rhizosphere sample. So this is what the plan would look like. What we're looking for is for farmers to take those samples in the untreated areas of their fields, the farm standard area of their field. So I think all of you are trying some sort of product or trying some sort of innovation in your field as part of the project. The samples that you'll be taking for the project will be in the untreated areas. That is so that we can get a consistency across the whole range of samples as well. And as I say, they should be taken from the same GPS location where possible. Please try to accurately map where you've taken your samples so that when you come back with your second rhizosphere sample, you'll be able to take them in the same location. And then the idea is, as I say, that we're looking to bring on the Shovelomics method, and we'll have some PES sensors this year as well that we'll be passing round to all the root ranges this year, so that you can assess your soil samples at the same time as assessing what the PES sensor says and what the Shovelomics reveals as well. So that's how you can get a better idea of what's going on in your treated area, is what we hope. And that's the dates that we're hoping to send the sample packs out, and the sampling period is from the 1st of March to the 30th of April. That's a little bit earlier than we had last year to make sure that you can actually get the soil cores into the ground. Now, I haven't really got enough time to talk about the specific protocols, but we can discuss areas that of concern at the end. However, you will get full guidance within the packs. And those who were involved last year, the feedback that we got was that those protocols were pretty good and sufficient at giving you the guidance on how to do it. But very happy to answer questions on that at the end. But I will just briefly say a quick highlight on 2026 trials. We have got 4 really interesting varieties that we're currently multiplying up that are going to be available next year for growers, and we'd like very much as many of you as possible, please, to try these varieties. 2 of them, those Watkins lines, have got biological nitrification inhibition. This has been tested in the field. They are the only varieties in the world that have that ability. And it's a really, really exciting and exclusive part of this project. You will be the first to put this fascinating new trait under test. We've also got Rossello and Capelli, which are 2 Durham wheat varieties, that have got really interesting rooting characteristics, and we want to explore, how these rooting characteristics differ from a normal wheat crop. I won't say any more because I've gone way over time, but so I'll just introduce Emma Chapelhow from UK Agritech Centre, a central part of the project. Emma, if you'd like to join us.
Morning, everyone. I'll just share my screen. Come on, buttons. Brilliant. Can everyone see that? Yeah.
Yeah.
Getting nods. Brilliant. So I'm Dr. Emma Chapelhow. I work at the UK Agritech Centre. We've been supporting the TOOF project. Very quick introduction to the UK Agritech Centre for those of you who haven't heard of us. We are an Innovate UK core funded team. There's about 200 of us now, and we support all projects kind of looking at accelerating agri-tech across the UK, as well as doing a few international projects. This covers everything from arable, horticulture, to all your livestock-based systems as well. We've kind of got experts across the field, including fish, which always surprises me, as like fish and seaweed is coming up. That's just a quick overview at the UK Agritech Centre, and I'll drop my email address in case you guys have any questions, or you may have seen some of our other projects floating around. So today, just doing a very quick kind of overview of the year 1 results, so you guys kind of see what it looks like and what you expect to see after this new year, and what it means and what we're kind of doing with that data as part of the project. So the first year results, what does this mean? We throw around a lot of numbers at you, with Maria doing metabarcode sequencing and Craig doing poor results and AMF fungi, but ultimately we're just trying to find out how healthy is our soil. And that's a question that's being asked of you, I think, probably daily at the moment. Everyone's preaching about healthy soil, how healthy is your soil, but as Tom mentioned, it's being able to measure it, having an accurate platform, and being able to kind of do that knowledge transfer between farmers to kind of understand what a healthy soil looks like on your farm. As we all appreciate, every farm is different, different soil types, different managements. So therefore, health looks very different on each farm. So to start off, we've got the University of Nottingham who are generating these weak core CT scans. So the CT scanning that you kind of imagine at the hospital when they go to look at your bones is what we're doing to your root cause. And as you can see, from this we can see the different things like the wheat roots, the wheat stem, earthworms. We can measure the soil pores, where there's stones, and this very cool video that will hopefully play again. We're able to create a 3D image of where the root structure is, which is the green image, followed by the air pores that are in the soil, and kind of monitor that complexity. Because these all have indicators of how healthy the soil is. So from this, we can take measurements such as porosity percentage, how complex that porosity is. We can map where the roots have gone. We can look at compaction. And what does this mean, I suppose, for soil health? It kind of indicates drainage capability, the water access for plant roots, how far the root can penetrate through the soil, kind of moving through that compaction and those compaction layers. It gives us an indication of microbial habitats, as different microbes live in different soil structures, and the pore structures gives us indication of compaction. It can tell us how efficient the gas exchange is through the different levels, and as well indicates nutrient transfer through the soil system into the plant. And jumping back to the microbial habitats element, and this then links really closely to what Maria at the John Innes Centre is looking at, and we'll jump onto that in a second. For the sake of this presentation, we've shown the porosity percentages across all the farms in our first year, and we've classified them loosely as organic, regen, and conventional. And you can see across the board a real difference of across all the farms, and the dotted line just under 8, so I think it was about 7.8%, is the average across all the farms. So as we build up the data year on year, we can really build out this image of what farms look like. We can start linking this to your soil characteristics or even the management techniques. As we can see here, we've done organic, regen, but at this stage, this graph doesn't really show any trends towards any of the management techniques. Is it going to go to the next slide? No? Okay. So yeah, as I mentioned, looking at soil microbial behaviors in the soil, we're then able to link this to what Dr. Maria Hernandez Serrano is doing at John Innes Centre. So she is looking at the soil microbiome. So what that means is she's collecting all the DNA from the soil, asking who is living here, what bacteria is there, what fungi is there, and what function are they performing. So to measure that, we look at the richness, so how many different type of microbes there are in the soil, and also how evenly are distributed, so their evenness. So are there loads of bacteria, are there loads of fungi, are there loads of nitrifying bacteria, are there less nitrifying bacteria, just how they're spread out. As I said, we're not just saying who's there, we're also interested in what they're doing, so we're able to link this to their functionality. So as I said, are they locking in nitrogen? Are they locking in organic material? Are they helping with that cycle? And this helps us understand the nutrient availability to the plant, how stable the ecosystem is, because that's a real sign of soil health, is whether the microbes are constantly fluctuating throughout the year or whether they're relatively stable and that they would be able to survive an extreme weather event. And finally, linking back, it links into this carbon. So how well is the soil able to lock in carbon? One of the other things Maria is doing is she's looking at the nitrification rate of your soils. And so this is the ability of it to convert ammonium into a nitrate, which is the form that the plant needs it in. And this process is driven by microbial activity to make sure that that cycle happens. And so this again tells us the nutrient cycling efficiency, so how readily available nitrogen is for your plants, whether the microbes are active. I often say that we have bacteria in the field. We know they're there. We know they're capable of converting ammonia into nitrogen. Are they actually performing that role? And that allows us to see whether they are actually doing the job that we think they're doing. And it again links to soil fertility and soil aeration because of that ability for gas exchange to happen. And then our next one is Dr. Tom Ferkel at the Crop Science Centre at Cambridge University. And he's looking into AMF, so arbuscular mycorrhizal fungi, and looking at who's there and then how many of them are there. So for those of you who don't know, I'll do a rundown. But mycorrhizal fungi are a beneficial fungi. They're often called the internet of the soil. You might have seen that floating around a few articles back in the day. And they help kind of connect plants to the soil. They communicate, they help with the transfer of nutrients, of water. And we're only really beginning to truly understand the function that they have. Every day we're learning more and more about them, but we're seeing them everywhere, and their roles are incredibly fundamental to this kind of communication between the soil, the environment, and the plant. So currently the feeling is the more arbuscules there are, means the more exchange there is between the plant and the fungi. So Tom's been identifying where the arbuscules are, and also how many there are. And again, we're seeing a real fluctuation across the different farm systems in our first year. But Tom said he's seen some of the most interesting things and some of the highest levels of AMF in our root range of soil. So we're really excited to see what's coming up in the next year and if we can repeat that. Just a quick shout out to the picture in the bottom there. You can see the root dyed blue, and the dark blue kind of stringy things are some of the filaments of the AMF fungi. And just, I suppose, to end, what we're now doing is bringing in all this data together. And I would just love to echo what Tom was saying. Like, we're doing a really amazing work in the Truth Project, and it really comes down to the root rangers and the work that you guys are doing and collecting all this amazing data for us. I think this is one of the most comprehensive soil studies I've ever seen, with all these different measurements and characteristics that we're testing. So, but again, at the center is the root rangers. We're looking at the different soil types, the different treatments, tillages, where it is, the variety. And then beyond that, we're able to test all these different things, really relate that back to the farm and the soil type, and hopefully create useful tools for the future. I think that's everything from me. So thank you. I just really want to thank all of you for getting involved in this amazing project. And I'll hand back over to Tom.
Great, thank you very much for that, Emma. That was a really great run through of the project and what we found out so far. And as Emma said, you know, we and those who were involved last year will know that there's an awful lot of data sitting there. We are only just beginning to understand that data. We've shared as much as we can at the moment, but there is more to understand, more to learn, more to share from that first year data that will be coming out over the next few months. And obviously the second year data will add to that as well. But I just would like to bring in Charlotte White now from ADAS, who's going to run through what we're going to be doing with Shovelomics. So Charlotte, over to you.
Thanks, Tom. I'll just share my screen as well. Hopefully that should come through as a proper screen share. Or can you still see it in slide view?
It's still currently in slide view.
Okay. There we go. That should work now.
You've got your notes on the side. I don't know whether you can switch that off.
Switch. Try that again, shall we? Sorry about that.
No worries. I noticed we've got there was a question that came through from Anna on the chat about whether this issue with the stones and the soil pores has been resolved. I don't think, unless you correct me otherwise, Emma, that is corrected yet. But we can. Has that been corrected yet?
Yeah, so I think it's one of the things, this is quite a new method of being able to get root cause from the live soil in the field, and we're always bound to have kind of methodological issues with it. I think as we get more cores in and we hit more stones, we'll be able to correct for that a little bit more. And there's definitely areas that we can manually extract from that data. So there's one core that we all saw where a big stone was right on the edge, and it kind of smushed all of the soil to one side and creating this giant gap. So in situations like that, we're really easily able to remove that. And then on that individual farm, because we have 5 cores going across the farms, we can kind of disregard that core to a certain element and look at the other ones and kind of take the average across the 4 and apply it. So I hope that answers a little bit, but as we get more cores, we'll be better at identifying where that has happened.
Has the actual data, the porosity data that you displayed earlier, I think that was the original data, so I don't think that takes into account the error, if you like, from the stones. Is that right?
No, that was from the original root ranging workshop in December last year. We're currently working with Maria and Craig to kind of bring the data back together again. So hopefully we'll get new graphs soon.
Great. Okay, I'll without further ado, I'll hand back over to Charlotte. Are you ready to go, Charlotte?
Yeah, I'm assuming it's all sharing properly now. It looks like it is from my end.
Yep.
Perfect, thank you. So I will give you a brief overview. I'm hoping my slides will change. There we go. So I wanted to give an overview of rooting and why it's important and why I find it really interesting, and then an introduction to shovelomics, and then focusing on the serial shovelomics protocol that we developed for the root rangers. So why measure roots? They are so important for every plant practically. And especially for crops. So they perform different roles at different points in the soil profile as well. So in the topsoil, they're really important for helping the seed to establish, for nutrient and water uptake, for providing anchorage so they don't lodge. They have those mycorrhizal soil bacteria associations that Emma and Tom have been speaking about, and they're also then combating any hard pans or compaction. Below that sort of 30 centimeter layer depth, they're involved in capturing water, especially later on in the season, uptaking soluble nutrients like nitrogen, and they're exploiting any cracks and biopores in the soil. And there are still those associations happening below down, deeper in the soil layers, but ultimately they are providing that uptake of water and nutrients, and really the only way the crop can access those resources. And roots are really difficult to measure due to how plastic they are, so how much they change in relation to local environments. And also the fact that the soil environment is really heterogeneous. It's very different in little places. There are pockets of nutrients or pores or stones, as we've seen, and it makes it a really, really difficult to measure soil roots, especially also because they're in the soil and we can't see them. But improving rooting, and the reason it's worthwhile measuring and digging up and having a look and persevering, is because improving rooting could help us meet current and future challenges for crop production. Improving rooting depth and vigor could help us improve water capture in drier climates or when we have drought, can help us beat soil compaction, can help us mine phosphorus fertilizer as supplies reduce, can help improve nutrient capture and therefore potentially reduce synthetic inputs. And they can also help us improve soil health and structure and fertility. So it's worth persevering, digging up all that soil and trying to have a look, but it's not the easiest thing to do. A little bit of background on serial root development. So serial roots have essentially 2 different types of root. They have the seminal or primary roots, which develop first from the seed. And typically there are 3 to 6 seminal roots that come from the seed at the beginning when the seed germinates. And this makes up about 5 to 10% of the total root volume of the mature crop. And it'll tend to be those seminal roots which will get deeper down in the soil layers, so they'll be taking up water later on in the season. After they develop, and once you get the nodal or crown or secondary roots, and these develop from the main stem and from the tillers, and there are typically 2 crown roots per tiller, but there can be up to 4 of them. And there are these really lovely diagrams on the side here. So these were drawn in 1926, where someone painstakingly excavated the soil around a root system and then sketched it out using a little grid. And that was done through the growth. And they're lovely diagrams, essentially, of loads of different crops and vegetable root growth, and you can get them on the internet if you're interested. And then I've also included here a picture of a barley plant where you can see clearly the nodal roots coming from the main stem and the seminal roots coming from the seed, and that little bit of stem that goes from the seed, the internode. So one of the questions is, how deep and how fast can roots grow? And they have different growth rates depending on the temperature of the soil, essentially. So when it's cold, there's about 6 millimeters of growth per day. And as the soil warms up into the spring, you can get root growth of up to 18 millimeters per day, which is quite a lot if you think about it. And root growth, so the weight and the length will proportionally grow during the active growing period, so through the foundation and construction phases in wheat. But then once you hit flowering, the root system is essentially at its maximum. You will continue to get root growth and some root death, so they maintain about that size throughout maturity. And roots can reach about 2 meters deep if the soil conditions and growing conditions allow. And on this graph, you can see that there are 2 critical root length densities, and root length density is the centimeter of root per cubic centimeter of soil. And 1 centimeter of root is required for uptake of water and mobile nutrients like nitrogen. But in the topsoil, to take up nutrients that aren't mobile, such as phosphorus, you'll need 5 centimeters of root per cubic centimeter of soil. But that's just it in the topsoil. And the majority of roots, so 60 to 80%, are also just found in that top 50% of your soil depth. So there's not that many roots when you think that below 50 centimeters that start doing all the water uptake to fuel the crop growth through to grain production. And these set of graphs, which are from the AHDB Wheat Growth Guide, they're really useful if you want to calculate how deep your roots are going for your crop, if you think about when you planted it and the growth stages and those growth rates through winter. And so you can get an average, an overall idea of what depth your roots might be reaching, which could be useful. We did some work looking at rooting in commercial crops. So what this graph has is across the top horizontally, you've got root length density. So that's the centimeter of root per cubic centimeter of soil. And you've got soil depth down to 1 meter. And then this is the critical root length density of 1 for taking up water and mobile nutrients. And I looked in the literature, and there's some really nice work looking at this in the 70s and 80s, looking at rooting to depth. And you can see that down to sort of 80 to 100 centimeters, the crops are very well rooted for water uptake. When we looked at 17 wheat crops that were assessed between 2007 and 2013, the average crop was not very well rooted for water uptake below 30 centimeters or 35 centimeters. And in the worst case, they were not very well rooted even in the topsoil. Thoughts as to why this could be happening in the change? the data from the 70s and 80s is a small data set, so that could be skewing it. But also we now have a lot more heavy machinery, which could be causing compaction. Soil is ploughed less, and there could be the potential of more modern varieties not having been selected for rooting in the same way. So and it could be a mixture of all of these to answer that question. But there's scope to improve rooting, especially at depth, to fuel yield by capturing water. So when we're talking about roots growing in soil, they have to use the tip to push their way through the soil to make a path. But they will also take the path of least resistance. So if there's a crack or a biopore, they will grow through this preferentially rather than having to use pressure to move the soil. And the optimal soil strength for root growth is 0.5 to 1.5 megapascals, and anything above 2 will start to restrict root growth and they won't be able to push their way through the soil. And this does differ for different species. Some are more able to push their way through than others. And there's been some work looking at where roots are found in the soil and in the soil profile, and there was some work done that found that below 60 centimeters depth, the majority, the vast majority of roots were found clumped together in these biopores and cracks. And what this effectively does is it restricts soil exploration. So the roots are limited to taking up whatever they can get in that little zone around that pore or crack. So it's important to try and improve our soil health and grow crops that will root deeply to try and improve this subsoil porosity and cracking, so that they can access more of the soil volume. So thinking about assessing rooting, one of the most very simple things that you can do is just take a spade out and dig up your roots and have a look. At a very, I say basic level, but it's not really a basic level, but less in depth than a full Shovelomics protocol, is a quick question of, does it look healthy? Are the roots white, or are they black? Do they look like they're growing strongly, or are they inhibited? Is there a pan that could be cutting them off, or do they look diseased and stunted? So that's a very quick health check. Thinking about the growth rates that I shared earlier, are they growing as quickly as you would have expected them to? And are enough roots being produced? So do you have sort of 3 to 6 coming from the seed? Do you have 2 to 4 from your main stem or your tillers? And then the check of like I said, is anything preventing the roots from penetrating deep into the soil? And that's something you can usually see quite easily if you just go and dig up and shake the soil off and have a look. I've included this diagram here because it shows really nicely the anatomy for a cereal plant. You can see the seed quite clearly, and you can see all the seminal roots coming from the seed. You've got the subcrown internode, which the seed produces, and it's usually the depth of about 2.5 centimeters, but it can be longer or shorter depending on how deep the seed's been sown. Then you can see the root crown, and the crown roots coming off the tillers and the main stem. It's a really lovely diagram, I think, for demonstrating that. So then moving on to Shovelomics and an introduction of Shovelomics. Shovelomics was developed in the United States by Jonathan Lynch in the Lynch lab, and it is a measure of the 3-dimensional architecture of the root system just in the topsoil, so we're not going to any real depth. And it was developed firstly in maize and common bean, and has now been adapted to look in cereals and other legume species. And it's been used to compare different varieties and genotypes to understand differences in rooting, to try and look for genotypes and lines that might root more deeply or root more widely, depending on the environment they're being bred for. And measuring roots, as I mentioned before, it's really time consuming. And if you want to go down to depth and look at how deep a crop's rooting, you have to take a soil core and then you have to wash it and then you have to pick the roots out and scan it, and it's really laborious and expensive work to do. So Shovelomics was developed to be more of a rapid and accessible method for quick phenotyping of genotypes, and to also then make those associations with more in-depth measurements to see if we can find something quick to phenotype those genotypes. And there have been some findings that a narrower root system that goes further down in the soil more quickly is related to deeper rooting at depth. So those species may access more water later on in the season, and a wider root system will explore more of the topsoil. So they may be accessing more of those immobile nutrients like phosphorus. So it's a really useful tool that lots of people are exploring and using in their research. And one of the main ways ADAS is using it at the moment is within the Root to Res project, which is short for Root Phenotyping and Genetic Improvement for Rotational Crops Resilient to Environmental Change. And it's a 5-year horizon project with the aim of delivering simple screening tools to facilitate breeders to look at root traits for resilience. So encouraging breeders to also dig deep and start using rooting to inform what they're breeding and the varieties they're bringing through. And we're also trying to identify some crop genotypes with idiotype traits, so ideal root systems to improve crop performance. We're looking at water availability, so drought and waterlogging, and also nutrient availability and the impact of nitrogen and phosphorus. And within the project, we're looking at 3 main crop types, but we're focusing on core crops of barley, potato, and fava bean. And if you want to know more there, you can look at the social media links down there or the website. But the reason I'm talking about Root to Res is because we have done a number of years of shovelomics measurements and related these to deep coring in the field. And so we've used this data to help inform the subset of shovelomics traits for the Root Ranger's shovelomics protocol. So a full shovelomics protocol has an awful lot of very in-depth traits to measure, such as branching, density, counting lots of different roots, and not all of them might be really useful for what we're looking at. And also it's an awful lot to ask of people who have an awful lot to do. So we have selected the traits that correlate with deep rooting and yield or other performance indicators based on 2 years of experimental trials that were conducted not only at ADAS in the UK, but also in France by Avalis and in Slovenia by KISS. So the traits that have been found to be consistent across all of those 3 sites over 2 years, we really believe are the important traits to be measuring. And so those are the ones that I've pulled out to measure for the Root Ranger's shovelomics protocol. And so to go into a little bit more depth for that, one of the other things I've asked of all of the Root Rangers is if you can also assess your plant population, because plant population can affect the root system and how it grows in the architecture. So if the plants are really close together within the row, they're more likely due to competition, to be more narrow and grow down, or to spread out more between the rows. So if you have a very dense plant population, that will affect the architecture compared to a less dense population. So it's important to capture the plant population. Also, will tell you how well your crop is established based on what you've sown. And it's preferably done early at the 2 to 3 leaf stage prior to tillering, just because it will make it easier to measure and the plants aren't overlapping. Given time constraints, I won't go into depth on this one, but I think you've all been sent an email that outlines how to do that. And if you've got questions, just get in touch and I can try and help. So the Root Ranger's shovelomics protocol is taken usually in the spring prior to stem extension, about growth stage 30, but technically it can be done any time. It's just best performed when the soil is moist and soft. And also if everyone's taking it at the same time, it's more easy to compare the results. You'll need a spade or a shovel from the Shovelomics, flag and a marker for labeling, a pen and paper to record your measurements. We will be sharing a protocol with you that will have what we're terming the Shovelomics board, to which records root system angle, some buckets and water for soaking the root systems, and a bit of soap, ruler, and some towels, paper toweling for drying. So in your area that you want to select from your farm standard treatments, we're looking at digging up 5 plants from 2 or 3 representative points. You'll need to dig up a few plants together, like you can see in this photo, because what you're aiming for is an undisturbed root crown. So if you start digging away from the plant or 2 plants that you're aiming for, 10 to 15 centimeters away, then you can prevent damaging those crown roots. And if you try and dig down to the full 20 centimeters depth around the plants, and you dig up the whole spit full, so you want the soil as well. And then once you've dug it up, you can remove the outside plants and allow any soil that falls away easily to fall away. And then you can put that in your bag, label it so you know which one it is, and then take it back to where you will be more comfortable washing the roots and taking measurements. So once you've got the plants back near a bucket with some soap, if you carefully remove the excess soil and put them in a bucket filled with water and a little bit of soap, which will just help to loosen up any clay particles, and leave them to soak for about 15 minutes. It might, depending on your soil type, you might not need to soak, or you might need to soak for longer than 15 minutes. And if you gently move them back and forth, and put your hands in just to try and get the soil off the roots, but not break the root system. And then if you leave them in water, I don't know if you get called away, if you leave them in the water rather than taking them out and letting the roots dry, it'll be easier to measure. And sometimes you can get 2 seeds that have been grown really closely together. And if you measure one of those plants that have 2 seeds really close together, you'll skew the results. So try and make sure that only one plant is being measured at a time. And it will be really nice for a visual capture if you could also take a photo before taking any measurements once the soil's been washed from the roots. And then measuring. So there are 1, 2, 3, 5 things we've asked you to measure. So first is the number of tillers, and that includes the main stem, because this will affect and is related to the number of secondary roots, crown roots you have. And then the width of the root system. So you can see these all marked in the photo here as well. So the width at the widest point. And then if you turn the plant around 90 degrees from the widest point, then you can also get the width of that point as well, and that will give the root spread in both directions, because the root system doesn't grow symmetrically. It will grow out into between the rows rather than intra row because of that plant competition. And then there's the depth to the maximum width from the shoot base. And then the maximum root depth which has been retrieved, and then the root system angle from the horizontal. So if you place, you'll have a copy of this board, and if you place the base of the root system in the center and then follow down and figure out which angle the root systems are growing, whether it's wider or narrower. So just to summarize, it's vital for crop growth to have a healthy root system. So it's important to check how your crops are growing, and therefore how well they're rooting, and if anything is inhibiting root growth. The root system I find it really fascinating. So and I think the more you look at them, the more interested you'll become. And for the Shovelomics protocol, we have a number of different measures that have been informed by trials and error. So we've tried to pick the traits that should really indicate how well the crop is performing and how deeply roots are growing. And then once you've got your measures and your photos, please share them with Boffin and your Boffin team. And thank you very much for your attention and for the opportunity to be in part of such an interesting project. And I'll hand over to Tom and stop sharing.
Great, thank you very much for that, Charlotte. That was a fantastic run through. I feel like I've learned a lot. Anyway, can't wait to get started. Look at my own crops, which I have to say are looking pretty appalling at the moment. So I don't think I'm going to find any great revelations. Just to say that we are preparing a full guidance on this, on what Charlotte was talking about. So there will be a full sort of protocol that will come out for you, as well as Charlotte was running through the different parameters that you'll be recording. So things to help you record that, to label up your plants and things like that as part of the pack that comes out to you, so that we can get some really good data from what you're measuring. And the really important aspect about this, though, is as you're doing this, get hold of your photos, post them up on the soil circle. Get the discussion going. Talk about what you're seeing. Ask questions as well. We'll be monitoring it. Charlotte will be there. She'll be able to respond to any replies. But this is a really, really important part of what we're doing. So any questions from anyone? First of all, either from Charlotte on her presentation on the Shovelomics, or from Emma on her presentation from the results. Any questions coming through from anybody?
No.
Oh, Anna, yes, got a question from you.
Sorry, question for Charlotte. When you're doing the root angle, surely you can just pull the roots to whatever angle you want, or is it not?
So the root crown will hold its structure generally. So yes, you could move them, but if once they're washed from the soil, they will tend to hold the angle they were at in the soil. So if you put them down and just try not to manipulate them too much, you should get a good idea of what the angle was in the soil.
Yeah, it's worth noting, by the way, that for those of you who are interested in the probity project that we've also got, that we've just recently started, we have got some gene edited. Well, they're actually, strictly speaking, tilling lines, as they're referred to, which have been edited so that they have a narrow rooting structure. We're going to be looking at that rooting structure within the probity project, but hopefully tying in the sorts of measurements that we're doing within this project to what we're doing in the probity project. So we get a really interesting idea, I think, of what the potential can be through manipulating genetics there. Also, another thing to note, Charlotte was talking about the plant populations. So within the Slimers project, and some of you are involved in that project, we are, and you'll know that you've been taking photographs of quadrats within your field, those people who have been involved. So all of those photos we're bringing together, and we are creating a tool that will, I hope, give you that we'll be delivering in an app, so that rather than having to painstakingly count all of your plants, you'll just be able to take a photo in the field, and it should be able to give you the analysis of that. So we're just working on that tool within the Slimers project. Any other questions from anybody else as we're going along?
Can I ask a question, Tom?
Sure.
Thank you. Sorry, as I'm quite new to the project, are you also looking at weather data at each of your root rangers' farms and fields, looking at rainfall, which will affect rooting, especially establishment?
I'm not sure we're actually gathering that data. I'm just.
I don't think we are actually.
Okay.
No, I mean, I guess it's data that is publicly available. Yeah. Why the question?
I just thinking about because I'm sorry, I've obviously missed the first year and how everything's coming together. And when I think about when you're growing a crop in the ground, when you can get it established will affect how it grows and how it roots, how wet the soil is, as well as if it was facing any drought during the growing period or when it was drilled, will also affect how it roots, and it will affect the soil structure and soil health as well. So I just wondered if any of the results were being put together in relation to how wet it was or how warm it was at the beginning of the season?
Yeah, I don't know whether the scientists involved, so University of Nottingham or John Innes Centre, are relating the results that we've got to weather data. We haven't been asking route rangers to capture weather data, and everyone has their own system of capturing it. So I'm not sure how we can get a consistent idea of weather data, but yeah, if that's something that we should be introducing, then maybe that's something that we can add into the data set.
Yeah, I wonder if anyone does have a weather station on farm they could, and they're willing to share whether it's very wet or very, I mean, we all know whether it's very, very wet or dry like last year, but locally it might be useful information just to explain some of the results, perhaps.
Yeah, we'll look into that and have a look at that, whether we can capture that and how we can capture it and bring it into the data. Are there any other questions or queries? It's quite a lot of information, I guess, to take in, and also I'm conscious that a lot of it is going to be on the protocols that you'll be receiving, and so there may well be other questions that you get that when you receive your protocols. And obviously, if there's any questions that you've got at any point, then just come through to Sky or to Laura or to myself, or preferably bring them onto the soil circle. If it's something for just that you want to contain within the group that you don't want to share with everyone else, the WhatsApp group is a great way to talk through, if you've got specific questions about the protocol, perhaps. But I just wanted to quickly share my screen, talk about a couple of events we've got coming up. Bear with me. So yes, just to we've got the workshop which we mentioned, which is at Will Oliver's farm. That's in Nuneaton in Leicestershire, and that's on the 18th of March. And what we'll be doing there is demonstrating shovelomics. We'll also be demonstrating the PES sensor, I believe, which we haven't talked about today, but is an integral part of this project. And as I say, we're going to have some PES sensors available for farmers to test their own soils as well, and then relate that to the microbial analysis data that they'll receive. And part of what we're doing within the project is calibrating that PES sensor. So that's taking place on the 18th of March, and it'd be great to see as many of you there as possible. You do get project ambassador points for all of these things, by the way. We're also going to be doing some boffin inspiration days. So these are going to be, we're planning to have, the plans are just coming together on this, by the way, so there's still quite a lot that we haven't finalized. But the plan is to have an event in the north and then the south, where we bring together, it's not just about truth, it's about all of our projects, bringing together people to, and it's a farmer-led event, where we really want to bring your ideas on how to develop the trials and how to develop the what we're learning from the trials as well. But they'll be hosted on one of our trialist farmers, I mean, if there's one of you, by the way, who'd quite like to host this event, then that would be fantastic. There would be an opportunity. What we're hoping to do is in the afternoon have a look around the trial, and in the morning it would be much more of a sort of workshop session. But we want to get together as many of our trialist projects across all of our trialist farmers across our projects. We have 70 trialist farmers at the moment, to bring them together to discuss in a workshop environment. There's also cereals coming up at Andy Ward's farm in Leadenham, Lincolnshire. And Boffin is a partner of cereals, we're delighted to say. So we're going to be very much involved in the webinar program. We have a stand there as well. We're going to have a little demonstration. For those of you who are involved in the Slimers project, you'll recognize the things that will be in the demonstration. Really, really want to see you there. And of course, there will be project ambassador points. We're going to have some food as well. We're not entirely sure whether it's going to be a breakfast or a lunch, on which day it's going to be, but we'll let you know anyway. So please do turn up to that. And of course, there's Groundswell coming up as well. And again, Boffin will be exhibiting there, and we really, really want to see you there. This is a great opportunity to talk about the Truth Project. We hopefully we're going to have some involvement in the seminar, the seminar programs as well. So really look forward to seeing you there. Now, unless we've got any other questions from anyone. I don't think there are any hands raised. Well, look, thank you very, very much for attending. We've taken a recording of this, so we will distribute a link to that. If you've got any questions at all, then do come back to us, either on the chat or come back individually to us. And also, please do take your thoughts and your observations, your photos and everything as the season progresses to the Soil Circle. The more you post, the more project points you will accrue. So thank you very much, everyone, for attending this. Really looking forward to seeing as many of you as possible over the various events that we're holding over the spring and summer. But it's now, I've run over time, so I shall let you go. Thank you very much, everybody, and goodbye.
Thank you.
Thanks.